everyone with a broad understanding of where we are. Last week, some of you in the front said, Pujita said that she quite enjoyed this lesson, Transcription Translation. We try to see if we have any doubts that we want to clear before we move forward anymore. Okay? This is one of the fundamentals of biology.
Here's a cell, all living tissue. Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in a nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, that's a very recap of transcription translation. Last week, a lot of questions were posed. Now I'm going to pose you back some questions. Right? If you want to discuss with your partner, please feel free to do so. We're going to try and make sense of it again. First question, where does transcription take place? Okay, feel free to talk. Ready? Okay, where does transcription take place? Nucleus. Okay, transcription takes place in the nucleus. What is the enzyme called that carries out transcription? Okay, so the enzyme is called RNA polymerase. If you want to, you can take out a textbook, flip to page 52 or 53. That's where we'll add transcription. Okay, so transcription, that's the first step. Transcription. The enzyme we're looking for is RNA polymerase. Where does RNA polymerase bind to before it begins transcription? It binds to the promoter. Promoter is a DNA sequence. Okay? It is not a block of protein, it is a double-stranded DNA, it is a sequence. Except that these codons are not meaningful. Okay? They are only a strand of sequence for the RNA polymerase to recognize and bind to. When I was in university, if I want to find a gene, I would type this sequence into the computer. Every time this sequence pops up, I know that to the side there will be a gene. So that's how I search for genes. I try to search for this sequence, the promoter. Flanking at the end of the promoter, you find a terminator, another sequence of DNA. It's not meaningful, it doesn't code for any amino acids, it's just for the RNA polymerase to recognize that it's time to terminate. It's time to stop my transcription process. Okay. So, RNA polymerase can do two things. The first one is it can unwind the DNA. The second, as the name suggests, it creates, polymerized means to create a strand, creates RNA. More specifically, it creates mRNA. A question for you next is, which strand do we use to create the mRNA? Okay, I hear the name template strand, but which is the template strand? Do I use the one on top or the one below? Depends on below. Okay, below, in this scenario below, our Q says it depends. Okay, let's work with below first, and then we'll come to why it depends. Okay, so if you look in your textbook, there's a phrase that says mRNA is synthesized in the what to what direction? 5' to 3' direction, right? Okay. So if you recall the chapter on DNA replication, every time we synthesize something, we synthesize in the 5 to 3. And we will read whatever we need to read as a copy from the 3 to 5. Okay? What do I mean by that? I'll show you in a while. Ivan was saying we're going to work the bottom strand. Let's take the bottom strand as a template. Okay? And now I'm going to synthesize a new strand. Let's begin first with A. Okay, what does it mean 5 to 3? This is the 5' end, the phosphate group. The 3' end is the hydroxide group. When I say I synthesize in the 5 to 3, it means I'm always adding to the 3 end. Okay, so let me find a U, put there. Okay, I'll put U here. I'm adding to the 3 end. This is me synthesizing in the 5 to 3 direction. I am reading the template in the 3 to 5 direction. Yeah? I read this in the 3 to 5, but I synthesize in the 5 to 3 direction. Follow? Okay, so let me synthesize it more. Can you help me out? So after this should be a G, okay. So one G and one C. Okay, let's go. A G and a C, another C. Okay, U. G, 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 C, and C. And here I've created a strand of mRNA. Okay, found it. CC. I've created mRNA strand, right? I just synthesized this in the 5 to 3 direction while reading the template in the 3 to 5 direction. Okay. So my question is, if we have to synthesize mRNA in the 5 to 3 direction, could I have not used this strand on top as the template? And then I just synthesize this way. See, this is 3 to 5, right? Okay. I'm still following the rules, right? I'm synthesizing now 5 to 3. Right? How do I synthesize it in this direction? Okay, but the promoter is here. Right? So, how does RNA polymerase know which strand I use as a template? Depends on where I find my promoter. Let me give you a scenario where the top strand will now be used as a promoter. Okay, imagine this was the promoter over here. Promoter. Right, now I know. Okay, I'm going to use the bottom strand. However, if my promoter was on this side, but read in the opposite way, promoter. Okay, let's say it's this way. Right now, promoter. If it's this way, that means I just now wrote the ATT whatever. You'll find it in the sequence here instead now. ATT, G, C, C. If it's read in this direction, the other strand will then be used as the template. So I realized this in university as I was typing in the promoter sequence. Then I was like, hey, how come I find some promoters in this direction, some upside down, and then some right side back up? And then I realized that actually it's a misconception to think that the entire DNA, one long strand, is where you find all the templates. The templates are found depending on how the promoter is arranged. Okay, this is for your information. That being said, if I ask you your class this question, a lot of students describe transcription as a photocopying process of the DNA. Okay? A lot of people say when you transcribe the mRNA, you are essentially photocopying the DNA. To what extent is this true? Okay? When you synthesize mRNA, you are photocopying the DNA. To what extent is this true? Okay, I'll give you a few seconds to discuss your favorite partner, then we'll see some responses. Alright? Okay, to what extent is this true? I just want to hear good things. I want to know the extent of the truth. Okay? Okay, fine. Who are the history students here? Okay, so you are to a certain extent. Now let's go for level 3 answer. Yeah? So to a certain extent. Okay, so to what extent is this true? Okay, the small extent. Wait, okay, why is this small extent? Yes, sir? Sorry? Okay, Sat says it is kind of photocopying because we are photocopying the non-template strand, except the U is, the T is replaced with U, right? To that extent, it's true. To what extent is it false? Okay, very large extent. What elements of my statement is false? DNA refers to the entire strand. Yeah, I might say DNA is double-stranded, right? So if we want to photocopy the double-stranded DNA, I should get a double-stranded DNA. Okay. Anything else that makes it false? Different letters. Okay. This is a U, so it's not exactly a photocopy. Anything else? Yes. Somebody tell me that this mRNA is way shorter than the DNA, so it's becoming a photocopy. Another person also say actually the point of reference is not the non-template strand. We actually make use of the template strand to create the mRNA. So we are creating a complementary copy in that sense, right? We're not taking the real thing to copy. I did not tell you, right? This strand is also called the coding strand. Does this have any meaning if I call it the coding strand? Yeah. I prefer to call it the coding strand because it has more meaning than calling it a non-template strand. If you refer to this ATG, AUG, but the sequence is essentially the same. That means to say you can use your genetic code to read the coding strand too, not just the mRNA. Okay? So you can use the genetic code to read both the coding strand and the mRNA. Which page is the genetic code at? 55. Okay. Now, let's see if it works. Can you read the coding strand and tell me what's the sequence of amino acids you should get? Met? Okay, met. Okay, first one. First codon is met. Second? Ala. Third? What? Met. Last one? Ala. Okay. Now let's look at the mRNA strand. First one? Met. Second one? Ala. Third one? Met. Fourth one, alanine, by the way, alanine methionine. So, the genetic code allows you to read both the coding strand and the mRNA. What it does not allow you to read is this. You try reading what TAC is, are you going to get methionine? Not CGG, you're not going to get alanine either. Okay? So that's what's the use of genetic code. A lot of students think you can only read the mRNA because there's only use there. You can actually read the genetic and the coding strands also. Okay? You can swap out the T's. So if you want to write down this little nugget of information, it can be helpful for you in the future. Okay? If I, for example, provide you with only the coding strand and this strand, could you find out what's the amino acid sequence without even writing out the mRNA? Yeah? It saves you a lot of time actually. Some students will go to the extent of, oh, I need to write out the mRNA first, then I can use the genetic code to read. You don't have to. You just need to read the coding strand because it's the same sequence as the mRNA. Okay, this information is powerful to help you read things fast. And when transcription is done, this is where we're going to go into the next thing. Okay, I close up my DNA. Now I'm left with my mRNA. Okay, I'd like you now to spend the next few minutes to do this activity. I'd like you to take a CA. Okay. I'll run you through the basic buttons and then I'll let you try it out. I'll work with a friend. It's more fun to see the outcomes of this. Okay. This simulation allows you to do two things. The first one is to transcribe. Okay, you'll find this button. Okay, so if you want to, how about everyone scan the QR code first, then you know what I'm talking about. Okay, activity 2 is the QR code you like to scan. You can find the QR code on page 1 of the class activity. Okay, QR code activity 2. Then you see this over here. If you are in, you will find several buttons. You will see transcribe, transcribe step by step, translate, translate step by step. Then there's a start, model, stop, reset, edit DNA. We're going to play with edit DNA. Okay? We're all very happy with transcription and translation. Now let's go into the next part of the lesson. What happens if we mess up the DNA? Okay? So that's what we're going to do today. We're going to mess it up. I'd like you to click on edit DNA and you will see a sequence. Okay? You click on edit DNA. Yeah? Alright. If you click edit DNA, the sequence you are looking at, the original sequence, that is this sequence over here, is referring to the coding strand. Okay, wait, I'm sorry, it's not sequence for sequence. I'm just saying that whatever you're looking at there is the coding strand. Okay? It is not the template strand, it is the coding strand. Follow? That means if you apply the genetic code on this, you can find out what the amino acids are. Okay? If you flip to the last page of your CA, you'll find a series of tables. Table 1 on the very top has this same coding strand sequence. The last page, table 1 contains the same sequence you find over here. Okay? Now, I would like you to click transcribe. After it transcribes, click translate. And I would like you to write down all the amino acids that are translated into the table. Okay, into table 1. Okay, I'm going to transcribe this sequence. This is what you should see. When you click transcribe. You should see the same thing, I think. Yeah? Okay. So you click transcribe, then you will watch the mRNA sequence being transcribed from the coding sequence that was shown in the just earlier on. Okay, so we'll transcribe whatever was given. Okay, Okay, and once you're done transcribing, you move on to translate. Okay, this simulator allows you to. Okay, then I'm going to go and translate. After you translate, you should get a polypeptide. You see the tRNA goes in. First amino acid is always methionine. You see peptide bonds are formed between all the amino acids during translation. Okay, step by step. The protein that I'm going to get is the result of whatever the original sequence is. Could you now write down all the amino acid sequence within table 1? So you see each table, the first row is the coding sequence, second row, you should write it in the amino acids that are translated. Okay? The second row is not for the template strand sequence, it is for the amino acid sequence. Okay, so everyone, you write down the amino acid sequence in table 1. Whatever this is, it starts off with methionine. Second one is choline, then glycine, glycine. Okay, if you play yours, you will be able to check the amino acids and fill it in. Yes. Can. Please write the short form like this. 3-letter word will do. Don't write out the whole thing. Okay? Just write out the amino acid sequence that you see. Oh, what is missing? Okay. What is the last one that's missing? Okay. Stop codon. You look to the genetic code. Is there an amino acid for it? Okay, so don't have that. So what happens is when the ribosome sees a stop codon, it just detaches itself. Okay? It doesn't translate it. So okay, should be okay, right? Okay. If you are still following me, this is where I'll let you do this yourself now. Now, let's go and edit the DNA. We are very happy with transcription and translation, but what if there are mistakes? What if the DNA has a little bit of a mistake? Click on Edit DNA. I'd like you to flip to the page of your CA. Step 7. Step 7. It says base pair substitution. What happens if I substitute certain base pairs for another? Okay? Can we look to the whiteboard first? When we say base pair substitution, we mean this. Imagine if I ask you to substitute this G of the coding strand. What I mean by base pair substitution is that I'm not only substituting the G with A, I substitute the corresponding pair that is supposed to pair it. So when we say a base pair substitution, we substitute the pair. Okay? That's why it's called base pair substitution. In your simulation, you will only need to edit the coding strand. Yeah? But understand that it comes as a pair. Alright, so can everyone try step 7? I'd like you to edit the DNA in that way. Base pair substitution. Can you substitute the G with a C? And then you go and translate that and see what you get. You understand? Okay, everyone, perform a substitution and then we will do a check of your amino acid sequence. Yeah? If you perform this substitution correctly, everyone should get the same sequence of amino acids. You can double check your friend, in fact. No, step 7. Edit the original DNA sequence. Right? By substituting the first C. Yeah. First C of the second codon. Second codon. Okay, you have to substitute the first C of the second codon, not the first codon. So if I highlight where you're supposed to substitute, is this fellow over here. Okay, you're supposed to substitute that with a what? With a G. Okay, substitute that with a G, and what do you? And then you go and translate it. Translate this. What do you get at the end of the day? Okay, alright. I would like you to write down the amino acid sequence. Okay, collect the data, compare with your friends. Have you all done the explore one? Okay, for those that did the activity beforehand, I have an activity for you. Compare all your products, all the sequences that you've done, to the original sequence. Okay? You should have filled up the last page, right? Okay, go and compare to the first sequence. Can you find similarities or differences? Okay? If there's anything similar, I would like you to highlight it to the original sequence. Meaning to say, did any of the amino acids turn out the same? Okay, I would like you to highlight it if you spot any. If there's one extra one, today, yeah, okay, I'm going to substitute my C with G. I'm going to apply it. When I click apply, I get something different. I'm going to check with you now. Do you get this amino acid sequence? Ready? Methionine, alanine, glycine, glycine, glutamate, serine, leucine, leucine. Oh, I don't know what I or E is. Glycine and leucine. You get that sequence? Okay. Now I'd like you to compare this sequence with the first sequence, the original one. Are there similarities? Are there differences in the product that you created? Where are the differences? Which codon resulted in a different amino acid? Codon 2? Okay, will you highlight that for me? Alright, we want to make as many comparisons to the original as possible. Okay, the aim for today is to see the effects of meddling with the original DNA. Here we have just performed what we call a substitution. And you find that wherever we substituted, we got a different amino acid, right? Okay. There's a name for such a mutation. I'm going to write here on the whiteboard. If you see that the amino acid has changed totally, we call this mutation a missense mutation. Okay, so that's why I'd like you to highlight that particular codon. To compare that codon to the original, it has changed. Okay, this is a what we call missense mutation. If the amino acid is different from the original. Yes. Sorry. Okay. Not all substitutions will result in a missense mutation. Okay. So what do I mean by that? I'd like you to look at the genetic code now. Is there a way that I could have substituted something for codon 2 but still get the same amino acid? Yeah. Notice each amino acid has a few codons that can code for it. If I get the same amino acid, we'll come to that in a while. There's a name for that kind of mutation. Okay, if you got the gist of how this activity is supposed to go, I would like to give you some time to complete the steps 8 and 9. How do you complete steps 8 and 9? The same idea. Click edit DNA. But before you edit the DNA, this is enough. Before you edit the DNA, I would like you to restore it to its original state. You substituted it with a G, right? Make sure you bring it back to a C, then you go and do your edit. Okay? So now I'd like to give you some time to complete steps 8 and steps 9. After you're done completing steps 8 and 9, compare to the original. Original. Find out what has changed. Alright? Have you found anything similar or different? Okay. Table number 3. Okay. So number 3, you found something similar to the original sequence, right? Okay. So leucine, is it? Okay. So there's actually a name for such a mutation where the genetic, the codon has changed, but the amino acid doesn't change. Okay, there's a name for this mutation. You can look in your textbook. There's a special name allocated to this mutation. Yeah. Oh, correct. Yeah, so that would be better. Correct. So actually, you can change the DNA sequence, but result in the same amino acid, right? Actually, it's as though there's no effect at all. Right? So we call such a mutation, so there's a name for it. When you find the name in your textbook, it has a lot of meaning. Yeah. Now you won't. So you will stop at the first one codon and encounters. And that's why I think your product will be much shorter. Do you get such a product? Oh, okay. So if you change the first one by right, you should get nothing. Yeah, but the simulation has limitations. Yeah, so you shouldn't get anything. Okay, once you are done with table 4, then you are free to explore. You can edit the genetic coding. You can edit the coding strand in any way you want. How do you edit your coding strand? So do you perform substitution? And do you get different amino acids? Okay. So you want to edit substitute every small. So in between, right, were there any amino acids that were the same as the okay, so GCC or you can so you identify what it means. So you call all the ones that change sensitive. You count how many sensitive. Okay, if you are done with table 3, I would like you now to count how many missense mutations you created. Okay, remember, a missense mutation is a mutation where the amino acid has totally changed from its original sequence. Can you count how many missense mutations you have? Okay, great. Highlight the missense mutation in a certain color. Right? How many missense mutations have you created? Okay, what I do is check carefully. The amino acid sequence is completely different. Okay, can you highlight the similarities and different colors? Yeah. You put stop. Yes. notice that the codon is correct. So my question is, suggest a reason why, suggest an advantage of having different codons code for the same amino acid. Why not just one codon for one amino acid? Why have a few for one amino acid? Yeah, so actually this is like a buffer. If I'm using one, right, it's not too bad. You know, there's a chance that I'm still getting the same amino acid. You need to spot all the distance. So you highlight this and this. So it's the same, right? Yes. Yeah, so actually, yeah, the ribosome just stays there. In fact, our protein won't get to eject itself, it should stay there. Here, everyone, count the number of missense mutations you created for table 3 and table 4. How many missense mutations do you create? To find out, you need to compare with the original amino acid sequence. You just disappear. What is it? So what you just done? You accidentally created the stop codon, right? For those of you that are doing table 4, do you get a shorter polypeptide? Okay, the question is why? Why it came out shorter? Okay, that is the question to answer. How do you create? Why do you create a shorter amino acid sequence? Oh, okay. So we'll highlight that. Okay, that's the key thing we've done. Because there are water molecules moving into it. So it just why is it? Okay. You Google? Yeah. There are many reasons. One of them is radiation. Yeah. Why is it? The precise mechanism I'm not clear, but if I'm not wrong, it could affect the way DNA is replicated. So it could affect the DNA replication process. Then rather than put in a certain base, accidentally put in another base. So mutations usually occur during DNA replication, where I'm supposed to put something in this sequence, but I accidentally put something else. Okay, now let's try to make sense of your observations. Okay, class, how many missense mutations did you create in table 3? Missense, huh? 9 or 8? 9? Okay, let's count up. So all the ones in blue are the missense. Are they 9? 1, 2, 3, 4, 5, 6, 7, 8, 9. Do you get 2 that didn't change? Okay. Notice that the codon has changed from the original, but the amino acid that got translated is the same, right? Okay. If you ever get such a scenario where the amino acid doesn't change, we call this form of a mutation a silent mutation. Make sense? Silent mutation? It's called silent because on the surface nothing has changed, but underlying it the bases have changed. Sorry? if it's a silent mutation. Okay, so if you ask, let's say your entire sequence just lots of silent mutations, would there be an effect on the protein? No effect. So that's why we call it silent mutation, because we don't know that anything has really happened. And in reality, we have a lot of silent mutations. Because if there is a problem, our body will fix the problem. But if our body cannot detect a problem, we carry a lot of these silent mutations. Okay? So we have a lot of proteins that are functional, but underlying it, something has changed.
Does it really effects on the audience?
No effects. Yeah, that's why it's silent. For table 4, you may have encountered this problem. Did you encounter a stop codon very early on? Yeah? Okay, such a mutation where you generate a stop codon is called nonsense. Okay, it's a nonsense mutation. This is nonsense. I haven't even finished my meal and you cut my receipt. Yeah, okay, so it's a nonsense mutation. Again, everything I highlighted in blue are missense mutations, as long as the amino acid has been changed. So I've introduced 3 kinds of genetic mutations that can take place. Missense, silent, and nonsense. The last mutation I would like to show you or describe to you is called frame shift mutation. Okay? You notice for the insertion and deletion, a lot of things change because now my frame of reference for reading the codon got changed. Right? Everything I was reading in triplets, but because I took out something or put something in, now my triplet, the way I read the triplet got changed. It's like the frame got shifted. Whenever you carry out insertion or deletion, frame shift mutation occurs. You shift the frame of reading. Okay, so 4 kinds of mutations I'm introducing here. You can shift the frame, you can get nonsense, you can get silent, you can get missense.
Missense means you change the codon.
Correct. Frame shift means that you shift the entire frame of your reading. Originally, there was one more code here, right? One more base over here, right? But because you deleted the base, everybody got shifted one frame up. Then all the amino acids got changed. Nonsense is if you create a stop. Okay, so frame shift is rather general. I can have a frame shift mutation along with many missense, along with a stop, along with a silent. So don't think of it as if you carry out a deletion, you will definitely get a certain mutation. I'm just showing the variety of mutations that can take place. Okay? So I say again, doesn't mean if you carry out deletion, you'll always get a stop codon being created. I've got instances where I insert and I get a stop codon. I'm just showing you the various types of mutation that can take place. Yeah, but frame shift will only occur if you insert or delete. Okay, is there a frame shift mutation if I insert 3 base pairs in?
If it's 3 different places, then if it's all
So a frame shift would be, right, if I insert 1 base pair in, right, and you throws off all the sequence downstream, right? But if I insert 3 in, actually, is it a frame shift? Right? Not exactly, because if I insert 3 in, everybody is not affected, right? It's just that I have one more amino acid in between now. So when we refer to frame shift mutation, we usually refer to our entire reading frame gets messed up. Yeah? So it's interesting, I insert 3, but usually the insertion is in 1 or 1. Yeah. If you insert 3, is it? Actually, I don't know. This was a thought experiment. Yeah. Not exactly right. You're not shifting your frame of reading. It's just one more amino acid. Yeah. If the tree put in a stop codon, then it's a nonsense immediately. Okay? So, to sum up, okay, I can substitute, I can delete, and I can insert, and it could generate this variety of mutations. Okay? You can therefore try when it comes to the explore stage. Go and try that. Go and insert, go and delete, you go and remove, see what you get. See if you can identify all the mistakes, all the stops, all the nonsense, all the silence. Yeah. Oh, okay, but you still got something. Okay, Shanae did this. She modified her start codon, but she still got a protein. Anyone do that? She modified her start codon. She replaced AUG with something else, but she still got a protein being shipped out. Okay, in reality, if she modified the start codon, you shouldn't get anything. But it's just that there's limitation to this simulation. It doesn't register that. So, by right, if you remove the start codon, or you modify start codon, you shouldn't get a protein at the end of the day. Okay? Yeah. Okay? In fact, something interesting, if you look at table 3, notice there's no stop codon. Right? So by right, the ribosome will come up to this point and just stay there. It will be hugging onto the mRNA and not letting go at all. Yeah? You need a stop codon to be able to generate a protein. So there's another limitation of this model. There's no stop codon, but it still managed to release the protein, which is not supposed to happen. There's no stop codon, the ribosome will hug onto the mRNA and not release anything. Okay? So there are a few limitations to this simulation. Okay, our time together is up. Is it chemistry ready? Okay, so I need you. Okay. If you replace, that's not frame shift. So frame shift occurs every when you shift the reading. Okay, that's all for today. Our next lesson is the quiz.